Close. Please close the door. شمرت ايديه.
Okay, hello again. Dear students, I'm Dr. Ranjbaran, Associate Professor of Physiology. We have 4 sessions in physiology together, and where are we from? Iran, India, Kashmir. Because I wanted to learn if I could use Arabic words. I know some Arabic words. For example, inshallah. Do you know what inshallah means? Okay,
لا يمكن بغضية.
four sessions.
نسمع إن شاء الله.
Four sessions, master physiology regarding tissue blood. I want to say something to you. Today we have class from okay, and tomorrow we have class from 13 to 15. Okay, my flight time. Okay, tomorrow my flight time is 14:30. Okay, I just follow them with the last session. Okay, what should we do? Can I continue today? Can I teach it today? Yes. Do you agree with me?
If you explain faster please.
Actually, I have a little daughter. Okay, for
بس
tomorrow.
الزرع حركة اليد.
Flight, the times for flight. Actually, were this time, and another one at 10 p.m. Okay, it was very late because my daughter is waiting for me. Okay, and there was no way for me. Buddy, for this reason, I want to cooperate with me. Can I teach it today?
We have a class after 3.
After 3, you have class.
Yeah.
Is it possible to continue our class today to 12:30, the second session, and after that the third session will start at 1 p.m. Do you agree?
والصلاة.
And tomorrow you have class from 8 to 12.
No, we have from 10.
And after that, can you come to the class tomorrow at 12? Can you come? At 12.
We have online class.
Very well. We have online class. 8 to
From 12 to
12. With Dr. Hassanzadeh.
Dr. Amin.
Dr. Mina.
What did you say?
These are past periods.
Are they updated or are they the same?
يعني كان 5 دقائق في المحاضرة وأنا طالب جودة أدرس.
قلت لك انه طفطف يعني
فترتك أحسن منك.
لا نحس لا نحل قصة السيشن.
I'm Dr. Ranjbaran, Associate Professor of Physiology. Regarding cell physiology, I'm going to start with the definition of physiology. Actually, what does it mean? Actually, physiology is a science in which we consider the organs in normal condition. We consider the mechanics under normal conditions. Okay. In our body, actually this 6% of body weight is water. Okay. 18% is for protein, 16% for fat. Actually, cell physiology is very important because it is a base, okay, basic subject for physiology 2/3 of water. 2/3 of water in our body actually is inside the cells. Another compartment is ECF. ECF stands for extracellular. Actually, it is 20% of this cell. Okay. And ECF in turn is distributed into compartments. One is plasma. From this amount, 5% of water is found in plasma. And interstitial fluid, IF, stands for interstitial fluid. From this amount, actually 15% is found in IF, interstitial fluid. You can see this in this slide. ICF, as you can see, is 40% of body weight, and ECF. ECF actually is composed of IF, interstitial fluid, 15%, and plasma, 5%. Any change in the composition of water, electrolyte, and so on, actually first is exerted in plasma. Why? Because all organs are in direct contact with plasma, not with IF, not with ICF. But any change, actually all organs, for example, kidneys, for example, GI, skin, lungs, all organs are in direct contact with plasma. Okay? Not with IF, interstitial fluid, and not with ICF. But I mean, the first changes are exerted in the plasma, and then after that, from plasma, the change is actually is transmitted. I cannot find the suitable word to other parts of our body. No, distribute is not beautiful. It's conduct. Here you can see plasma. Now we are going to talk about electrolyte composition. Okay, in plasma, in IF, and in ICF, ECF, and ICF. But as you can see here, the concentration of ions in plasma and IF are the same. The concentration are the same. Why? Because this membrane is the membrane of capillaries, okay? Because the membrane of capillaries is very permeable. It allows to ions to move freely between plasma and IF, interstitial fluid. But if I ask regarding the cause of having the same concentration, okay, between plasma and IF, don't forget, it is as a result of existence of permeable capillary wall. Permeability of capillaries is high for this reason. Ions can move with ease. No questions? You have to memorize. In cell physiology, you have to memorize that. Suppose that it is one cell. Don't forget. All the time, sodium concentration is greater outside the cell. What does it mean? It means we have no sodium. means lower concentration. Body, you have to memorize it. Not need to memorize the values of, for example, sodium concentration is ICF and ECF. That's enough, you know. Sodium concentration is greater outside the cell. Okay, bicarbonate concentration is greater outside the cell. You have low amount of bicarbonate inside the cell. Chloride concentration is greater outside the cell. It means we have low concentration of chloride ions inside the cell. The story about potassium ions is completely different. Potassium ions actually are found in higher concentration inside the cell. Okay? And lower concentration outside the cell. The story is the same for phosphate and proteins. Only phosphates and proteins with negative charges are accumulated inside the cell. Okay? What does it mean? It means cell membrane has no permeability to proteins and phosphate. But for this reason, proteins and phosphate with negative charges remain inside the cell. Got it? No question. Please not go ahead. If you have any question, don't hesitate to ask. If not, please not go ahead. Okay? Give a feedback to me. Okay? Give a reaction. Show a reaction. Got it? Thank you. As you can see, sodium concentration is greater in ECF rather than ICF. As we know. For example, potassium ions. Pay attention to potassium ions. Potassium concentration in ECF is low. In ICF, it is high. As I told you, phosphate proteins are found in higher concentration inside the cell. What's the cause of this difference? What's the cause of these differences in concentration of ions across the actually between ICF and ECF? It depends on the cell membrane. Cell membrane is actually has a great, is not permeable, sorry, is not permeable to ions. Okay? Is not actually as permeable as the membrane of capillary wall. But you can see permeability here, but here there is no permeability. If I ask you regarding the cause of this difference between ions in ECF and ICF, the cause is plasma membrane. Actually, plasma membrane doesn't allow ions to move freely. No question.
Okay. Wow.
Sure.
Here you can see the values regarding ions. As I told you, not to memorize it. Okay? As you can see, sodium concentration is greater outside. Potassium is greater inside. Calcium ions outside. Is greater outside. Chloride ion outside. Bicarbonate ion outside. And you can see pH and osmolality that we will talk about that in renal physiology, not in cell physiology. No question. Was it clear up to here?
Yes.
Regarding homeostasis, cell physiology. In physiology, actually, we have two similar words. One is homeostasis. Second one is hemostasis. Hemostasis is constant condition in external environment. In internal environment, sorry. Internal environment. Internal environment. What does it mean? It is the same as ECF, not okay, I might raise a question regarding the definition of homeostasis. Okay, tell me which one is correct. Maintenance of nearly constant condition in ICF. Maintenance of nearly constant condition in ECF. Maintenance of nearly constant condition in internal environment.
True.
2 and 3 are correct. But no question. Actually, every factor, everything in our body under normal condition must be in a normal range. Everything has a normal range in our body, such as blood pressure, for example, pH, sodium concentration, potassium concentration, glucose concentration, and everything, even membrane fluidity. But in everything in our body actually is regulated in a normal range. Beyond this range, below this range, actually it is not physiology. But what is maintaining in normal range is called homeostasis. In homeostasis, actually, we assume that body acts as a home, and our body is like a home. The stability of this home must be maintained. For this reason, we say homeostasis. It is the origin of homeostasis word. Suppose that we eat something. What happens after eating? Potassium concentration greater, actually is greater more than range. For example, glucose concentration increases more than normal range. The body is not in homeostatic range. What should we do? Actually, the body tries in healthy people, okay? The body tries to return back this amount, back to normal state. But it is called actually homeostasis. What's the reaction of body when potassium increases more than normal range? Body tries to decrease or increase? Decrease it. Body tries to decrease potassium. It is a stimulus. It is response. Glucose concentration increases. Body tries to decrease. Now suppose that blood pressure, a stimulus is decreasing in blood pressure. What's the reaction of body? Body tries to increase blood pressure. Okay. Suppose that when blood pressure decreases, body decreases and decreases it more and more. What happens? It leads to death. Suppose that when potassium increases, body increases it more and more. What happens?
That's
Yes. Glucose concentration decreases, for example, and body decreases it more and more. It leads to this. Okay, for this reason, we say in our body, most of the control systems, control center, act by negative feedback. What does it mean? It means the direction of the stimulus is opposite to the direction of response. It is called negative feedback. Okay, in these conditions, actually, positive feedback leads to this. Got it? No question? question. Was it clear up to here? Actually, most of the control systems act on negative feedback. Components of a control system. I told you all factors, every factor in our body is regulated in a normal range by a control system. Okay, in physiology, you will get familiar with thousands of control systems. Correct? The component. The first component is receptors. Actually, receptors sense the changes. For example, when glucose decreases, what happens receptor? Actually, sense this condition. Okay. When, for example, blood pressure decreases, receptor sense such a condition. The second component is what's the reaction of receptors? Receptors send signals to a control center. What's the location of this control center? Just actually, all organs are control centers. But not correct. Every organ might be a control center. For example, in regulation of glucose concentration, pancreas is control center. But we will talk about that. I will take some examples. Okay. What's the reaction of control center? Actually, it sends some signals, to effectors. And effectors return the for example, a factor back to the normal range. But let me take an example regarding glucose concentration. Glucose concentration. Suppose that glucose concentration increases. Receptors in this example are in pancreas. Receptors in the pancreas actually sense such a change. Okay? But they send signal to the control center. Control center in this example is pancreas 2. Okay? What's the reaction of pancreas? Pancreas release insulin. Insulin release. And insulin affects on some effectors. Effectors here are, for example, fat and muscle tissues. For example, liver. Liver cell might be another effect. Why? Actually, insulin allows fat, muscle tissues, and liver cells in order to remove excess glucose from the blood. Now, glucose concentration returns back to the normal range. Got it? Another example regarding, for example, blood pressure. Suppose that blood pressure increases. Okay. Suppose that blood pressure increases. As soon as it increases in blood, get familiar with parasympathetic in heart physiology,
Come on.
cardiovascular block. Okay.
Go on.
Actually, bioreceptors send signal to the control center. Control center here is brainstem. Brainstem, basal. Motor system, brainstem. Brainstem actually sends signal to the blood vessels, and the heart. Actually sending signal to blood vessels causes the blood vessels become dilated in order to decrease blood pressure. Okay? So we have vasodilation. We have vasodilation. How about the heart? How about the heart?
لا
Actually, cardiac output increases or decreases.
What you do is
Do you want cardiac output is? Cardiac output increases. But the cardiac output, CO stands for cardiac and by its mechanisms, blood pressure decreases. Body
حلو يعني.
if body stasis in homeostasis, there is no risk, there is no problem. In circular, actually all the time, our control systems are regulating all factors. But if body fail to maintain homeostasis, what happens? Certainly, we have this body here on here.
No mercy, right?
What are you laughing at? Actually, these are introduction to cell physiology. Actually, the first session is introduction to cell physiology. Okay, this slide is regarding homeostasis. Here, as you can see, there is problem in homeostasis, and here, as you can see, actually, can I see the cursor? Here, homeostasis is established. But in a patient who is not able to maintain homeostasis,
That's
in this slide you can see the components of the control system. in order to close puncture site, in order to stop bleeding. Okay. Actually, here we have positive feedback in the second step. How? Actually, activated platelets call other platelets, other inactivated platelets to come to the puncture site. It is a positive feedback. This positive feedback continues until actually bleeding stops. Body. Suppose that the puncture site is great and is large and definite blood formation is not able to close puncture site. What happens in the third step? In the third step we have blood clot formation. In blood here, platelets are involved here, a lot of factors, a lot of proteins inside the plasma are involved. A lot of factors become activated and they begin to activate other inactivated factors. It is an example of positive feedback. Okay? Positive feedback is seen in the step 2 and step 3. And step 4 is regarding repair of injury site. Positive feedback or seen in step 2 and step 3. Ready? Blood formation and lipid blood formation. This is the first example of positive feedback. The second one. The second one is normal vaginal delivery. Okay. Suppose that it is uterine and this part is cervix. My painting is awful. Okay. And head of baby. But it is head of baby and this part is cervix. Okay. During delivery, what happens? Actually, the head of baby exerts pressure to the receptors. Receptors of cervix. It is an example of control center, which acts by positive feedback. Okay? Receptors are in cervix. Receptors send signal to the brain. Here, in this example, brain is control center. What's the reaction of brain? Brain releases oxytocin. OCT stands for oxytocin. And oxytocin affects some effector. What is effector here in this example? Effector is the muscle in uterine body, not in cervix. In cervix we have no muscle, okay? In uterine body, in order to contract stronger and stronger. And this part, again, we have actually more pressure here. Release of more oxytocin, stronger contraction. But it is positive feedback continues until the baby is born. Got it? The third example is action potential. AP stands for action potential. We will talk about this part in more detail. But let me talk in brief. In action potential, during action potential, voltage gated sodium channels open. You have to name it completely, name channels completely. Okay? Voltage gated sodium channels open, and it causes more and more voltage gated sodium channels to open. And action potential actually is elicited and is propagated by this positive feedback. We will talk about that in more detail. No question? Okay, let's go to the cell membrane. Are you familiar with the structure of membrane? Yes. Alhamdulillah. Some students, okay, say that you. Very good. Actually, cell membrane is not physiology, but we have to talk about that. In market this way, from lipids, proteins, and carbohydrates, sugar. Okay. Different types of lipid. The most abundant lipid in the membrane is phospholipids. Another type is glycolipids. And the last one is cholesterol. Proteins. Protein in the membrane are integral or peripheral. Okay. We are going to start with phospholipids. You are familiar with the structure of phospholipids. Actually, phospholipids have a phosphate head, and two tails. And this part is head. Head is hydrophilic. What does it mean? It likes water. It likes to make connection with water. But tails are hydrophobic. They escape from water, but they don't like to make connection with. Suppose that I have a container with water. Okay, and I put some phospholipids here. Head of phospholipids are in direct contact with water, and the tails escape from water. But in the cell, in our body, in ECF, we have a lot of water. In ICF, we have water. What should we do? Actually, head of phospholipids are in direct contact with water in ECF, ICF, and the tails here. Okay? In ECF, heads are in contact with water in ECF, and the tails are escaping from water. For this reason, cell membrane is composed of lipid bilayer. It is one layer, okay? We say lipid bilayer. Because we have two layers of phospholipids. But it is one cell membrane. For example, we say mitochondria have two membranes. What does it mean? It means here, this membrane is composed of lipid bilayer. This membrane is composed of lipid bilayer too. Got it?
Yes.
Very good.
Okay.
Is the base of cell membrane structure. Regarding glycolipids, glycolipids actually in glycolipids, sugar part is phospholipids, alcohol and phosphate pair form hydrophilic region. Two tails form function of membrane. Actually, in a bacterial cell, we have just one cell membrane, one lipid bilayer in this actually, which forms cell membrane. What does it mean? It means it has no organelles, such as mitochondria, such as endoplasmic reticulum, and so on. For this reason, in a bacterium, we have just one cell membrane, one membrane, which is called plasma membrane or cell membrane. What's the function of cell membrane? The main function, it has a lot of functions. Okay? The main one is that it doesn't allow ions, water to move freely. Okay, as I told you in the previous slides. Again, what in eukaryotic cells? We have a lot of organelles inside the cell. What does it mean? It means we have a lot of membranes inside the cell, plus cell membrane or plasma membrane. Actually, everywhere we see membrane, it means we have concentration difference across that membrane. But for example, here you can see one organelle. As you can see, the concentration inside this organelle is completely differently. The concentration in plasma. But the concentration in plasma is completely differently. Concentration is this. No question. It is the main function of cell membrane. Membrane. Other functions. Receiving information. Receiving information by which part? The virus. All cells have some receptors based on the type of cells. Okay? Receiving information. The second one is import and export of small molecules. I told you it makes cell membrane makes a barrier. It doesn't allow actually water electrons to move freely. Okay? And the third one is capacity for movement and expansion. No question actually. Movement is important, for example, for during embedded life, cells must migrate. And for example, in an adult person, it is important for blood vessels. When they have to move from the blood, for example, to tissues. No question. Aren't you tired? May I keep teaching you? May I keep teaching you? Or do you want to take a break? Please be energetic. I try to make cell physiology actually very easy for you. Actually, cell physiology is difficult, okay? But I try to make it. I make sure. I make it sure. I try to make it easy. Regarding the movement, actually all lipids, all types of lipids in the membrane are in constant motion, are not static. The membrane is a dynamic motion. Okay. This motion might be, for example, flexion. You can see here, for example, in this part, the tails must be, might be, for example, it is called flexion of the tails. Okay. Another movement is rotation. Okay. A molecule for itself, it rotates. Another one is actually lateral motion, lateral movement. Okay, for example, this molecule actually want to move in this direction. Another one is flip-flop. It means moving from one lipid to the other. But it moving from, for example, this one is called flip-flop. Let's talk about the role of cholesterol in the membrane, and you can take a look. Okay? Role of cholesterol in the membrane. The most abundant lipid in the membrane is phospholipid. It is must be regulated in a normal range. Suppose that a sliding is in this way. In this way we have unwanted pores. And we don't want to have unwanted pores in the membrane. But for this reason, amount of motility must be maintained in a normal range. It is called homeostasis. Actually, cholesterol plays major role in this regard. How actually we say cholesterol? When cholesterol is distributed in high concentration in the membrane, it decreases motility. It decreases fluidity. Motility of membrane is called fluidity. Okay? When cholesterol concentration is great in the membrane, it decreases motility. It decreases fluidity. Okay? But on the other hand, we don't want to have a solid membrane. Okay? All cells, for example, all static. We don't want to have solidity. In this case, cholesterol is removed from the membrane and fluidity is regulated in a normal range. But if you forget actually the function of cholesterol, all the time I take an example for students. Cholesterol in the membrane is actually large with lower in the soup, for example, in the soup. What does it mean? Suppose that you are preparing soup, okay, and now it's time to eat. But the problem is that our soup is watery. Okay, it means fluidity is great. What should we do? One way is adding with lower soup. It absorbs excess water. Okay? And actually it is the same as cholesterol in the membrane. One student told me it means cholesterol absorbs excess water in the membrane. No. Okay? Cholesterol decreases or increases. Cholesterol regulates the fluidity of the membrane. But it was just one example. No question. Take 10 minutes break. Take 10 minutes is enough.
Let's finish this PDF and then
You want to actually, I think some students are taking a nap.
you can say it. It's easy, honestly. There's like
10 minutes
pages.
is enough. It's 15.
There's
10
7
minutes.
pages.
10 minutes. See you at 9:25. Buddy, 9:20. Please be on time. Buddy, they need to membrane retention and so on. Okay. I'm going to focus on integral proteins. Integral proteins in where? Inside the cell, outside the cell, in the membrane. Okay? When we say integral protein means proteins in the membrane. They have different functions. They might be receptors. In endocrine physiology, you deal with a lot of receptors. In cell physiology, it is not our focusing point. But they might be enzyme. They might be anchor. What does it mean? It means by this protein, cell membrane itself attaches to, for example, cell wall, attaches to extracellular matrix, attaches to cytoskeleton, and so on. And they might be transporters. Our focusing point in cell physiology is the last function of integral protein. Actually, we have one integral protein with different function. For example, one integral protein might be a transporter and at the same time it might be a receptor. We will talk about that, such as ligand-gated channels. We will talk about that. One integral protein might be a transporter and at the same time an enzyme, such as sodium potassium pump. We will talk about that, okay? Some integral proteins have just one bond, some have actually samples. Kadi, two or more bonds. Kadi, no question, you have to memorize it. Transporters. Transporters, I will focusing on these transporters. Transporters might be channels. I need more space. We are talking about integral protein in the membrane. Got it? Transporters might be channels or carriers. How can we identify one protein is channel or a carrier? Okay. Shape of channel is in this shape actually. In the channel, you can see a pore. It is pore. It is a channel. But we actually draw carrier in this shape, you cannot see a pore. Okay? Got it? Channel, channel. Channels are leak channels, CH stands for channels, or gated channels. Leak channel has another name, simple channel. Leak channel or simple channel. Okay. Leak channels have no gate. What does it mean? They have a hole which all the time is open. These channels are open all the time. Whether electrolytes can move based on the size. Based on the size of where? Size of the smallest, the narrowest part of the channel. But the electrolytes larger than this amount cannot pass. But the positive, we say these leak channels allows water and electrolytes to move based on size. However, for example, suppose that we have a molecule in this size. It cannot pass. Got it? Sometimes a lead channel have negative charges because of amino acids. Okay? This channel allows water and positive ions, which are smaller than the size of channel to pass. You see, these channels allows movement of water on electrolyte based on size and charge. Because it is covered by negative charges. These negative charges actually don't allow negative ions to pass. It repels negative ions. Study this channel actually allows movement based on size and charge. Sometimes, actually, some lead channels have positive charges. Say this channel allows movement based on size and charge too. It doesn't allow positive charges to pass through this. It repels positive charges. It allows water and electrolyte, which electrolytes actually negative ions, which are smaller than its size to pass. Kadi, no question. Kadi, it is very important. It is based on action potential, based on resting membrane potential. If you have any question, don't hesitate to ask. Try to go ahead with the class, okay?
Does each channel have both of the charges, positive and negative?
No, we don't have a channel with both of gated channels. One type is voltage gated channels. Don't forget, in action potential, we are talking about voltage, changing in voltage. It means voltage gated channels are involved in action potential. They are very important in AP, action potential. But what does it mean? We have a gated channel. For example, it is multi-gated sodium channel. Multi-gated sodium channel has two gates. One in this part, one in this part. Okay? When voltage changes across the membrane in enough amount, okay, this gate is open, allows ions to move freely. Because voltage gated sodium channel, actually it allows sodium ions to come inside. Okay. Another type is ligand gated channels. This side again, we have a gated channel. We have a gated channel with a site for a ligand, with a receptive site. In this example, as you can see, it is ligand gated channel is transporter and is receptor too. Why? Because it has a receptive site. Active after ligand binding, what happens? This gate opens. A ligand must be bound here in order to open the gates. But after opening, ions and molecules can. No question. No question. Example, acetylcholine gated channel in neuromuscular junction. We will talk about them in more detail. It's a type of ligand gated channel. Okay. The last category is regarding storage or pressure. Storage or pressure gated channel. Pressure gated channels. Actually, in nervous system physiology, you deal with storage gated channel and special senses. We have storage gated channels. Have you heard about Pacinian receptors in our skin? Pacinian receptors for pressure in our skin.
Yes.
Let me talk about that. In nervous system physiology, you will study in more detail. Pacinian receptors are pressure receptor in our skin. Okay? It is composed of, it is the structure of receptors, some capsules. Okay? These capsules are filled with a special fluid. Okay? We have a special fluid in these capsules. Got it? In the center of this capsule, there is actually sensory part of a neuron. Okay. In the membrane of neuron, we have a stretch. In the membrane of this neuron, we have a stretch. Gated channels. Let me show a stretch gated channel in an expanded view. I want to draw one is pressure or stretch gated channel. It is gated channels. It means it has gate. The gate of this channel is actually attaches the membrane by some filaments. Okay? Repeat one. What happens when there is a pressure here and pressure is exerted here? What happens? Here, the membrane is actually, the pressure is exerted, is applied to the membrane. As a result of this pressure, these filaments are pulled And after fluid, what happens? The gate is open and allows ions to move inside or leave the cell. But when there is a pressure here, I told you these are pressure, receptor for pressure. There is a deformation in the shape of capsule. This deformation exerted pressure by fluid. I told you here we have a special fluid. Exerted the pressure to the next capsule. Again, we have deformation here. Fluid applies a pressure here. Again, we have deformation here. And this pressure is applied to the membrane. By this pressure, actually these filaments are pulled, gate is open, and it doesn't lead to action potential. It might lead to an action potential. We will talk about that. These are different types of gated channels. We haven't talked about simple diffusion, but don't forget. When a channel is open, either big or gated channel, don't forget. Water, not water. Actually, electrolytes move and molecules move based on simple diffusion. What is simple diffusion? Moving from higher concentration to lower concentration. Don't forget. When we say, for example, the flow in this channel is in which direction, you have to say it is done based on simple diffusion. Again, don't forget. Channels, simple diffusion. Any types of channel. Okay, what carriers? Carriers, we have two types of carriers. As I told you, in carriers, we cannot see a pole. They have two types of carriers. Carriers which are involved in, facilitated diffusion. D stands for diffusion. Carriers involving in active transport. Active transport. Channel is for simple diffusion. Carriers are for facilitated diffusion and active transport. For carriers, actually carriers are transporters and receptors through. I mean, carriers have receptive sites in internal or external part of the protein after binding ions or, for example, glucose. What happens? There is a conformational change of the facilitated and active transport. Again, no question. You have to memorize it as there is no other way. You have to memorize it. Carrier for what?
Carrier function.
Carrier function.
Carrier
protein
Function. Some carriers are involved in facilitating the diffusion. Some are involved in active transport. When we actually describe about active transport and facilitated diffusion, you can actually find it. Okay, no question. You know what electrolyte means? What is electrolyte? So they try to memorize transport. The transporters are channels or carriers. Channels, big channel, gated channels. Carriers, facilitated diffusion in active transport. Okay, that's enough. You know, that's for today. Substances in our body. Substances are considered in two categories. Fat-soluble substances, water soluble. Alright. Regarding fat soluble, we are focusing on movement across the membrane. For this reason, I have to draw membrane. Suppose that in this membrane we have phospholipids and, for example, a lichen. That is fat soluble molecules such as oxygen, carbon dioxide, nitrogen. These are fat soluble. What does it mean? It means for oxygen transport from carbon dioxide. Actually, we don't need any channel, any carrier, and so on. They are fat soluble. For this reason, they move with these oxygen based on pressure gradient, carbon dioxide based on pressure gradient. Oxygen from inside the cell, carbon dioxide leaves the cell. For fat soluble molecules, we have no problem. Why? Because they pass the phospholipid layer, the lipid bilayer with these. The only point regarding fat soluble molecules is that I don't say fat soluble ions. These are molecules. These are not ions. Ions are water soluble. Okay. Fat soluble molecules. The only point is that as their size increases, the speed of transportation actually decreases. Okay? As their size increases, the rate of diffusion decreases because they move slowly when they are large. Kadi, no question. Regarding water soluble, we have two types of water soluble. One is molecules, such as glucose, and second is ions. Ions are called electrolytes. Electrolytes is the same as ions. Why do we say electrolyte? Because they can conduct electricity. For this reason, ions can conduct electricity. For this reason, we say they are electrolytes. They have two types of ions, positive and negative. Kali, no question, don't say. They have to memorize it. No question? Nothing to study this one. Nothing to study. In this slide, you can see the different functions of integral proteins in the membrane. As I told you, our focusing point is transporters. Actually, this figure shows that proteins in the membrane can move to exactly the same as lipids in the membrane. But the movement is limited. Movement of proteins is limited in the membrane. What is some mechanism? One mechanism is here. You can see integral proteins are in contact with cytoskeleton. Actually, it limits the movement of proteins. This attachment limits the movement of proteins. Here, integral proteins are in contact with extracellular matrix. This attachment in turn decreases the amount of motility. Proteins are able to move inside the membrane, but this motility is limited by some factors. One factor. Second, as you can see here in the third line, integral proteins from two adjacent cells are connected together, are attached together, and by this way, they might be gap junctions. Okay? And might not be gap junctions. Okay? This attachment doesn't allow protein, for example, this protein to move and come here. Okay? This attachment doesn't. And this one, it is very important in physiology, renal and GI they are absorptive surfaces. Renal and GI system, they are absorptive surfaces. The cells in the actually in GI system and in the kidneys are connected together by tight junction. Here is luminal tubes in the kidney, luminal GI, very luminal, for example, intestine, epithelial cells, and here is interstitial fluid and capillaries. Okay. Epithelial cells are connected together by tight junctions. Type of proteins. Okay. For this reason, proteins in this side of membrane is called luminal or apical membrane. Apical membrane. The membrane which is in direct contact with lumen is called luminal or apical membrane. This part of membrane is called basolateral membrane. Basolateral membrane. Body. Proteins in luminal membrane can move in this region between two tight junctions. Why? Because these tight junctions actually don't allow proteins of luminal membrane to come to the basolateral body. And a protein here in the basolateral membrane can move in this region, And it cannot come here in the luminal membrane. Why? Because tight junction doesn't work. Okay? Suppose that, for example, glucose is supposed to be removed from glucose. Here we have a specialized transporter for glucose absorption, only absorption in the kidneys. And here glucose must move in this direction. Suppose that this protein moves and comes here. What happens? Glucose is excreted, and we don't want to have such a thing. But these proteins of luminal membrane, must be in this part of membrane, and they cannot come here. And this protein in basolateral membrane must move in this region, not in the luminal membrane. Got it? You can see it. Again, tight junction, luminal membrane, and basolateral. Got it? And the last one is regarding the role of sugars in the membrane. As you can see, sugars in the membrane are found just in external part of the membrane. In the internal part, we don't have sugar in the membrane. Okay? Just in the external part, they might be in direct contact with proteins. In this case, they are called glycoproteins. They might be in direct contact with lipids. They are called glycolipids. Okay? Section of the different functions of different roles of actually sugars in the membrane. Okay? They have a lot of roles. No question? Okay, the first session is over. Was it clear?
Yes.
Really? Are you energetic to continue to give teaching? Transport mechanism across the membrane. In this session, we are going to talk about transport mechanism across the membrane. Okay. Here you can see different transport mechanisms across the membrane. Okay. First is simple diffusion. Simple diffusion. Second one is osmosis. Third one is facilitated diffusion. The fourth is active transport. Endocytosis, exocytosis, solvent trap, bulk fusion. The words in red, they are active transport, endocytosis and exocytosis. It means these for molecule is ultrafiltration. They don't need energy as ATP. For simple diffusion, for facilitated diffusion, for osmosis, we don't need energy as ATP. Buddy, no question? And as I told you, you have to memorize that sodium concentration is greater in which substance? In outside. You have to memorize it, okay? Because in simple diffusion, we want to guess sodium moves in which direction. A channel is open, okay? I told you in channel, when a channel is open, ions, molecules move based on simple diffusion, okay? We want to guess sodium moves in which direction. For this reason, we have to know sodium concentration is greater outside. Chloride concentration is greater outside. Bicarbonate concentration outside. Calcium concentration outside. Potassium concentration inside. Proteins like phosphate, sulfate, got it? Glucose concentration is greater outside. What does it mean? I told you for fat-soluble molecules we have no problem. They move based on simple diffusion from higher concentration to lower concentration. For example, oxygen and carbon dioxide. Both are moving based on, for gases, we say they move based on pressure gradient, not concentration gradient. Okay? They move from higher pressure media to lower pressure. Got it? But here, these are different colors, different shapes, means different water-soluble molecules and ions. Okay? My molecules and ions cannot pass through lipid bilayer. Why? Because they are not fat-soluble. They are water-soluble. Okay? To solve this problem, membrane is equipped with integral proteins. Actually, the integral proteins are transporters, not anchor, not receptors, not enzymes, okay, transporters. And the membrane provides a pathway for moving based on size and sometimes based on size and charge. When channel inside the channel has charge. Got it? Regarding the movement, suppose that here we have a lot of sodium ions, okay, and that's outside. There is actually low concentration of sodium. What happens? Sodium ions here, think about that. We are more here. It's better we move in this direction. It is moving based on, actually based on thinking. It's not correct about molecules and ions. Okay? Actually, they move randomly. Molecules and ions move randomly based on kinetic energy. Okay, they have kinetic energy. Here, we have a lot of sodium ions. Ions and molecules are in constant motion, which is called kinetic energy. Got it? A lot of sodium are moving here. All the time they strike with each other. They strike with each other and they change their route. Okay? Got it? In this way. In this side, we have a lot of sodium. It moves, we have a lot of strike here. For this reason, at each moment, more molecules begin to move in this direction. From higher concentration to lower concentration. Got it? In this side we have 4 sodium ions, for example, 4 sodium ions. These ions move in this, for example, direction. And here, there is one strike. It changes the route. Here, there is another strike. It changes the route. Again, it changes the route. At each moment, low molecules, low ions move in this direction. Okay, this movement is not done in a smart way. done randomly. Okay, it is the concept of simple diffusion. Okay, simple diffusion is moving from high for molecules. Simple diffusion for molecules and ions are different. For molecules, we say simple diffusion is moving from higher concentration to lower concentration. What does it mean? If, for example, here we have a lot of glucose molecules, we are talking about molecules. A lot of glucose molecules, low concentration of glucose molecules. Okay. What does it mean? Simple diffusion is moving from higher concentration to lower concentration. What does it mean? It means no molecule, no glucose molecule move in this direction. It doesn't mean no glucose molecule move in this direction. We say movement in this direction is greater, is higher, but it is the concept of simple diffusion. No question? Okay. It is concept of simple diffusion for molecules, but for ions. Actually, this figure shows actually striking of molecules together and changing the path. Ready? As we can see here, in area A, concentration of molecule is great, in B is less. Diffusion from A to B is in this direction and in this amount. Diffusion from B to A is in this amount and in this direction. For this reason, in simple diffusion, net rate of diffusion is important. Okay, net diffusion is important. Net diffusion is in this amount, less than this one. Why? Because some molecules are moving in this direction. In this amount and in this direction. Got it? Suppose that what happens? After a while, we have the same concentration of molecules in both sides, A and B. What happens? Molecules remain constant. Molecules are in constant motion. Okay, all the time they strike each other and they change their route. But moving from diffusion from A to B is in this direction, in this amount. Diffusion from B to A is in this amount, in the same amount, and in this direction, in the opposite direction. For this reason, we say no net diffusion. But it means molecules and ions are in constant motion. No question. Regarding simple diffusion. Simple diffusion for molecules. A shows simple diffusion for molecules. As you can see, net diffusion is from outside to inside for that molecule in panel A. Okay. In panel B, I told you we are talking about ions. For molecules, concentration gradient or chemical gradient determine the rate of diffusion. Let me repeat it. For molecules, concentration or chemical. Chemical is the same as concentration. Both are the same. Concentration or chemical gradient. Or another name for gradient is driving force. Driving force. Concentration or chemical gradient determine the movement, the direction of movement. For ions. For ions, we want to take a break. Wash your face, dear boys. Wash your face if you are not good. May I continue? But do remember, I told you inside the membrane, we have protein accumulation. Do remember with negative charges. Phosphate ion again with negative charges. Sulfate ion with negative charges. And low amount of, for example, bicarbonate. Low amount of chloride ions with negative charges. Actually, cell membrane is not permeable to proteins, phosphate and sulfate. For this reason, they remain inside the cell, and inside the cell outside because of presence of proteins, sulfate and phosphate, which actually that membrane is not permeable to the body, but we have permeability for chloride ions or bicarbonate ions. Okay? For this reason, inside the cell is more negative than outside. Okay, it is very important. Be careful. Inside the cell, we have a lot of positive and negative charges. Sodium ions, potassium ions. Okay? What's the fate of them? Actually, in the center of cell, they begin to neutralize one another. But the excess negative charges, which are not neutralized, accumulate immediately under the membrane. What happens in extracellular fluid? In extracellular fluid, positive and negative ions begin to neutralize one another. Excess positive charges accumulate here, adjacent the membrane. Why? Because they want to attract one another, but cell membrane doesn't allow. Right? They want to attract one another, but cell membrane doesn't allow. For this reason, inside the cell membrane, we have excess negative charge. Outside, we have excess positive charge. When a cell is at resting the cell, means when there is no, for example, action potential. Okay. Was it clear?
Yes.
Have we learned about capacity?
Yes.
In physics. Actually, membrane is a capacitor. Why? Because it stores negative and positive charges. Okay? Membrane acts as a capacitor. Okay, not need to memorize it. It's not actually for medical students. Got it? Inside the cell, I told you. The cells inside the cell, we have a lot of positive and negative charges. They begin to neutralize one another. Okay, positive and negative charges actually are placed next to one another. Excess negative charges accumulate under the membrane. Outside the cell, it is the same actually. We have the same rule. Positive and negative ions begin to neutralize one another. Excess positive charges accumulate here. Why here? Because they want to attract one another. But cell membrane doesn't allow. Okay? For suppose that in this condition, I want to write this point. We have a membrane with excess negative charge inside the cell, excess positive charge outside the cell. Sodium concentration is greater outside and is lower inside the cell. Okay? Suppose that we have a channel which is permeable to sodium ion. Okay? What happens? Based on concentration or chemical gradient, pay attention please. Based on chemical and concentration gradient, sodium moves in which direction? Chemical or concentration gradient is moving from higher concentration to lower concentration. Based on chemical or concentration gradient, sodium moves in this direction. I'll write CH. CH means chemical gradient or concentration gradient. Okay. For ions, this negativity and this positivity effect on diffusion, on movement. Actually, negativity inside the cell. This negativity, actually negative charges cannot leave the cell. They have to remain inside the cell. Why? Because I told you the membrane is not permeable to them. What happens? They cannot leave the driving force, net driving force for sodium is in this amount, greater than electrical and chemical gradient, and in this direction from outside to inside. If we have a channel, sodium likes to move inside the cell with a great driving force. But for molecules, concentration or chemical gradient is actually determines the amount and direction of movement. For ions, electrochemical gradient, G gradients or electrochemical driving force determines the amount and the direction of movement. Got it? Tell me regarding potassium ions. Potassium concentration is greater inside. Inside the cell is more negative than outside. Tell me please, based on chemical gradient, chemical or concentration gradient, potassium moves in which direction? Outside to inside. Chemical means moving from higher concentration to lower concentration. It is moving based on chemical gradient. Body, based on electrical gradient, potassium moves inside to outside.
Outside.
Actually, this negative charge doesn't allow potassium ions to leave the cell, and at the center, it wants to attract more positive ions. Electrical is in this direction. Don't forget, under physiology condition, I mean in a live person, all the time, electrical gradient is greater than chemical gradient in our body. For this reason, all the time, potassium likes to leave the cell. Here, chemical and electrical are in the opposite direction. For this reason, net driving force for potassium is less than this amount and in this direction. Got it? Was it clear?
So, meaning that potassium ions don't leave. They cannot leave the cell.
Doesn't leave the cell. They cannot leave the cell.
No, I'm asking, can they leave the cell or do not leave the cell?
Actually, under. Let me say this sentence at the end of this session. Here in this example.
If it is more positive charge to more potassium, it can be, but in this condition cannot.
The natural negativity inside the cell doesn't allow positive ions to leave the cell.
Put more potassium.
You are talking about which driving force? Electrical or chemical?
Electrical.
Electrical actual negativity inside the cell determines electrical gradient in this example. Negativity doesn't allow positive ions to leave the cell. And at the center, they want to attract, pull positive ions from ACF to IC. Got it? Was it clear? Dear students, no problem? What's the problem? And net driving force is sum of electrical and chemical gradient together. Okay, let me take another example. Okay. Another example. Suppose that we have a cell with higher sodium concentration. This one, inside the cell is positive. Is it possible to have positivity inside the cell? During action potential, we have positivity inside the cell. Tell me, based on chemical gradient, sodium moves outside to inside, chemical gradient. Based on electrical gradient, how about electrical gradient? Positivity don't like to positive ions to come inside. For this reason, electrical is in this direction. But don't forget, net driving force is in this direction. direction. Why? Because chemical is greater than electrical all the time. In a live person, chemical is greater than electrical. It is during action potential. During resting. During resting, let me show it again here in order to conquer them. During resting, the state inside is negative. Chemical gradient is in this direction. Electrical is in this direction. Net driving force is in this and in this direction. Got it? Got it? Now, we want to talk about potassium, concentration, chemical and electrical, right? During resting state and during action potential. What happens? Chemical gradient in this example is in this direction. Electrical is in this direction during resting. Net driving force is in this direction and it's small, it is small. But here, chemical gradient is in this direction. Electrical gradient is actually this positivity plus positive ions. Okay? Electrical is in this direction. During action potential, pay attention to the net driving force of potassium ions. It is greater than that of the resting state. Ready? Let me check with my sense. Under physiology condition, all the time, under either resting or action condition, all the time potassium likes to leave the same. ICL to EC. ICL to EC. Right. Sodium, ECl to IC. ECl to IC. But sodium under normal condition, either at resting state or during action condition. Sodium likes to come outside. Potassium likes to leave the site. But the amount of net driving force is different. Tabii, no question. Actually, action potential just to sodium and potassium ions are important. For this reason, we don't want to talk about bicarbonate ions, for example, fluoride ions. Yes.
During action potential, the net driving force.
During action potential
Yes.
for potassium.
Yes. The net driving force.
Net driving force. Chemical gradient is greater. Actually, both are in the same
Yes, the
direction.
same direction.
For this reason, net driving force is greater than chemical and electrical gradient individually. Okay? No question? For molecules, we say molecules move based on chemical or concentration. For ions, we say ions move based on electrochemical gradient. Actually, negativity and positivity of inside the cell has no effect on molecules because molecules have no charge. Right? We have another driving force for simple diffusion. It is moving based on pressure gradient. It is used for gases, as I told you, for oxygen and carbon dioxide. We say oxygen, carbon dioxide, nitrogen, and other gases move based on pressure gradient. It means from higher pressure to lower pressure. It is simple diffusion. Okay, in cell physiology, just we deal with electrochemical gradient and chemical gradient. But in cardiovascular physiology and renal physiology, you deal with pressure gradient. Simple diffusion based on pressure gradient. Okay, keep it in your mind. Pressure gradient for renal physiology. Actually, I will teach renal physiology to you in China. You have no way to escape from me. All semester, I will be here in China. we have tried to talk about this one. Simple diffusion. Not need to memorize formula. Okay. But tell me please. What's the actually effect of concentration gradient of substance on simple diffusion? Suppose that we have more concentration gradient. More concentration gradient leads to less simple diffusion or more simple diffusion? Surface area. Suppose that we have a great surface area of membrane. Great surface area of membrane leads to more simple diffusion or less? More. Because the possibility of having a lot of channel for ions and water soluble molecules increases in the membrane when we have a great surface of membrane. But the third one. Lipid soluble. Lipid solubility, the third one is for fat soluble molecules. When lipid solubility increases, the rate of simple diffusion increases for fat soluble molecules, not water soluble. The fourth one is molecular weight. Suppose that one molecule is large, one molecule is small. Which one moves actually is small? Well, actually molecular weight has negative effect. There is a negative correlation between molecular weight and rate of diffusion. And the last one is distance. Distance is thickness of the membrane. Okay, thickness of membrane is composed of a lipid bilayer, cell membrane. And the thickness is something about 7 nanometer. It is the same 7 nanometer. In all cells, the thickness is the same. Why do we have this factor here in cell physiology? It is for myelinated fibers. By myelination, are you familiar with myelinated fibers? By myelination, the thickness of membrane increases. Got it? For this reason, the rate of diffusion increases. Don't forget. It means in myelinated region, in myelinated fibers, simple diffusion decreases. Actually, we need it. It is very essential in propagation of action potential. Not need to memorize the formula. Okay? These factors have negative, positive correlation. These factors have negative correlation. Not need to memorize the formula. Okay? What does it say? It says in simple diffusion, as concentration of substance increases, rate of simple diffusion increases properly. Buddy? We have talked about lead channels and gating channels. You are familiar. Don't forget, simple diffusion for fat soluble molecules is done through lipid bilayer. For water molecules, soluble is done through channels. Either lead channels or gated channels. We have simple diffusion. Okay? If I ask you regarding, for example, I draw a gated channel and I say sodium moves in which direction. Okay, you have to know. It is based on simple diffusion. Okay? Different types of gated channels are shown here. Actually, a stimulus here for opening up these gated channels is membrane potential. It means it is voltage gated channel. Okay. The second, actually, we have a gated channel with response to external ligands. We have a channel with response to a messenger, intracellular messenger. And the fourth is intracellular metabolite. 2, 3, and 4 are ligand gated channels. Okay. And 5 and 5 is esterase gated channels or pressure gated channels. No question? Was it clear? Don't you have any question regarding simple diffusion? Simple diffusion is over, alhamdulillah. May I keep teaching?
Yes.
Osmosis. What is osmosis? Osmosis is very important for renal physiology. It is very important. Try to keep it in your mind. Try to internalize in your mind. Okay? What is the definition of osmosis? Osmosis is simple diffusion of water. What does it mean? It means moving of water molecules from higher concentration to lower concentration. What does it mean? Suppose that we have two containers. Osmosis is simple diffusion of water. I have two containers. Be careful with this, okay? I have two containers. They are the same. Inside the container, we have the same volume of a solution. But the solution is the same in container A and B. Here we have water and some dissolved molecule and ions. And in B container B, we have this amount of 3 molecules or ions. Molecules and ions are this. Tell me please, in which container concentration of dissolved molecule is greater? In A or in B? In A, concentration of dissolved molecule is greater. In which container concentration of water is greater? Right? Because the volume of the in B, concentration of water is greater. Molecule here is greater. Okay. Now suppose that I have a container and I divide this container into 2 compartments by a semi-permeable membrane. The volume of water is the same into volume of solution is the same in A, compartment A and compartment B. Pay attention, please. It is very important. Suppose that here I have some dissolved molecules. What happens here? We have osmosis or simple diffusion of molecules. Which one? Bravo.
بعد ما اصور لو تودع
Are dominant. Now, this time I fill this container with the solution. I divide it into compartment A. In compartment A, we have a water and dissolved molecule of ions. In compartment B, we have water and low concentration of water. What is dominant process here? Osmosis is dominant. Osmosis from A to B or B to A? B to water molecules comes in this. Okay, it is prerequisite for osmosis. Membrane is not permeable to molecules and ions. Water moves. Why? Because water is the smallest molecule in our body. All channels are permeable to water. But lipid bilayer is not permeable to water. All channels in the membrane are permeable to body. It is the prerequisite for osmosis. When there is no permeability for ions and molecules, osmosis is dominant process. Got it? Actually, in this example, in that example, osmosis never stops. Why? have the same volume of solution here. A semi-permeable membrane, A and B. Here we have pure water. Here we have water and molecules actually, and there is no permeability to molecules. Again, membrane is not permeable to these molecules. What happens? Water moves from B to A. Oh my God. Water moves from B to A based on osmosis. Water comes to A, comes. What happens as a result of water entering? Volume of solution in A increases. At the same time in B, volume decreases. Osmosis continues until the pressure of this column as a result of difference in the surface of water. The pressure of this column of water, actually the pressure is exerted to the membrane. Okay? It continues until the pressure of this column doesn't allow water molecules to come inside. In this condition, osmosis stops. It is called osmotic dispersion. Pressure of this difference in pressure is called osmotic pressure. Osmotic pressure actually stops osmosis. And I described just one slide and you can take the question. No question. Thank you. We say in osmosis, actually we have a formula in physics. It is kinetic energy K equals M V to the power of M divided by 2. It is 2. A or in B?
What?
In A. Because actually concentrating gradient in B is less than that of in A. In A, osmosis is greater. For this reason, we say osmosis depends on the number of molecules, not mass, not velocity. Correct? You're very smart. I believe in you. Take a 10-minute break. After that,